Hello everyone, this is our second lesson in Unit 3. We're still talking about DNA, we're still talking about genetic information, but now we're thinking about how new genetic information is formed within cells in the same organism. So we're not talking about making sex cells, like in human sperm and egg. We're talking about just copying DNA, right? Which is going to be important for making those sperm and egg cells, but we'll talk about that later this year, not right now. So right now we're going to talk about DNA replication. DNA replication is done semi-conservatively. So this means that when DNA is replicated, when it's copied, 50% is going to be old DNA and 50% is going to be added to it. So in this diagram, I really like it, we've got one double helix of DNA over here. This would be our first generation, right? And you can see all the DNA is colored blue. When it replicates, it's going to split, and we're going to have one strand up here and one strand down here. These would be called daughter strands. So daughter strand up here is 50% blue. That came from the original copy. And 50% is this yellow. Now that 50% yellow has been added to it. Right? And because of Chargaff's rules, those base pairing, right, adenine to thymine, cytosine to guanine, it's going to have the same information. But we're still thinking 50% old, 50% new, right? Now the second daughter cell is going to do the same thing, right? 50% came from up here, and then 50%, that yellow part, was added to it using Chargaff's rules. Adenine, thymine, cytosine, guanine. Now remember, the cell isn't like reading the instruction manual and being like, oh yeah, this is an adenine, so we've got to staple a thymine here. Right? It's looking at how many hydrogen bonds it has. If it has 2 or if it's got 3 hydrogen bonds. Right? But each of these daughter cells is going to eventually replicate its DNA. So that same blue piece of DNA is going to end up in this daughter cell here. But this one is going to be 50% new and 50% from this one. Again, this yellow strand here is going to end up here. This blue strand is going to end up down here. And same thing down here. So this is done through DNA replication. Here are the 3 basic steps of DNA replication I want you to know. All right, I'll let you write this down and then we'll talk about it. So hit pause, write all this down, and we'll talk about it again. So to replicate the DNA, first it has to be uncoiled. So that parent strand is all twisted up in a ladder form. It's going to have something called helicase. Helicase is an enzyme. Remember, anything that ends with ASE is going to be an enzyme. It's going to unwind the DNA. You might hear this referenced as unzipping the DNA. All right. So then something called polymerase is going to come in and match new nucleotides to the existing strands. In your textbook, there was a great animation of this if you watched it. If you didn't, I encourage you to go back and watch it. Very useful. But new nucleotides are brought in, matching with those old nucleotides. Adenine to thymine, cytosine to guanine. All right? And then termination is where it's coiled back up, and there's also some error checking that goes on. The error checking process is something that scientists are actually still studying, trying to figure out exactly how that works, because if we can maybe improve that, or we can look at how the body checks for those errors, we might be able to check for genetic diseases or cancers much better than we are right now. All right, so here's a diagram of this. I am not going to ask you to draw this in your notes, but I do think it will be very helpful for you to have in your notes. All right, so not asking you to draw this as part of the assignment, but I think it would be a good idea. All right, what we do see here is the unzipping and unwinding of DNA. All right, so over here on this right side, we have the regular parent strand of DNA. Right. Topoisomerase is an enzyme that just straightens it out. That's all it does. It unbends it so that it's straight. Then we have helicase here, which looks like this blue triangle. That's not the actual shape, just saying that's what it is here. Helicase is breaking it apart into what we call the leading strand and the lagging strand. Now these are called this because they move in different directions. your DNA has some molecules on the end of it that are either 5 of them or 3 of them. We call these the 5' end and the 3' end. Right? So we're always going to be having a directionality to this. All right, with the leading strand, DNA polymerase is going to come in and add the nucleotides to match using Chargaff's rules, right? And connecting the sugar phosphate backbone all at the same time. Right? This is the easy one. The lagging strand is where it gets a little bit more complicated, all right? But for now, we're going to kind of think about this as something pretty easy, right? This is our leading strand. The lagging strand, we have to go a little bit differently because DNA polymerase is going to be traveling in the opposite direction. Right? So we have primase coming in and putting little markers on so that RNA primers know where to go. Right? And they bring in these nucleotides. The Okazaki fragments are just groups of the nucleotides, and then DNA ligase is kind of gluing everything together and connecting that sugar phosphate backbone. All right? So DNA replication always begins with the enzyme DNA helicase. It breaks those bonds between complementary bases in the DNA and exposes the bases inside the molecule so they can be read by DNA polymerase. And that's used to build 2 new DNA strands with complementary bases. Complementary here means like opposite, A to T, C to G, complementary. All right? The 2 daughter molecules that result, each molecule of DNA, contain 1 strand from the parent molecule, 1 new strand that's complementary to it. As a result, the 2 daughter molecules are identical to the parent molecule. All right? So they are exact copies. No mutations is the goal. Right? DNA replication is semi-conservative because half of the parent DNA molecule is conserved, meaning still present, right, in each of the 2 daughter DNA molecules. Right? I do want you to try to think of 2 questions that you could look into to either fully explain this whole process step by step or the semi-conservative part. Right? So think of 2 questions for the end of your notes. And I'm going to check those tomorrow.